Hi, everyone. Thank you so much for joining us today. My name is Justin. I'm a clinical researcher for CureCMD. I'm very excited to be moderating this session today. We're going to be talking about SELENON research, and we have a couple panelists here to talk about the projects that they've been working on. Our audience members have asked a couple questions that we hope to get to. We also hope to have some time at the end to answer some questions live if you guys have any. Before we get started, there is a disclaimer statement that I do need to share with all of you. So I'll just go ahead and briefly share my screen. So the information and opinions shared by our panelists will be generalized statements and are not intended to be interpreted as a promise for future treatment outcomes or a specific timeline for such outcomes. While we are eager for advances, it is impossible for anyone to predict when a treatment may become available or anticipate roadblocks. This session is being recorded and will be shared on our public YouTube channel after the conference portal closes on August 31st. So now that we've gotten that out of the way, why don't we take a moment to go around our panelists and have everybody introduce their name, institution, and their discipline.
Not sure what order you'd like us to go in, but I'll start, I guess. I'm Alan Beggs. I'm at Boston Children's Hospital. I'm a molecular biologist by training and have been, as some of you know, very interested and focused on SELENON myopathy and related congenital myopathies for many years. How about
Anna?
So I'm Annette Ferreiro. I'm a neuromuscular neurologist and scientist. And well, together with Behzad, we've been involved in the very first descriptions of this selenoprotein and related disease. So it's very close to my heart. And well, we're working both on understanding the mechanisms that lead from the mutations to the signs in patients and in finding treatments, particularly drug treatments, and also in care and clinical aspects of the disease.
Since you mentioned Behzad's name.
Yeah. So hello, everyone. My name is Behzad Moghadaszadeh. I'm a scientist, molecular biologist by training. And as Anna said, this project is very close to my heart. This was my thesis project when I was a grad student in Paris. And with Anna, we were working side by side on the first description of this disease. And since then, I have come to Boston Children's Hospital, and I work with Alan Beggs on this project. So we have a number of people actually in the lab working on the SELENON disease, and we are approaching. We have taken different approaches, gene therapy and drug screening, and we have also a mouse model and a zebrafish model to try all these different approaches.
Esther.
Hello, everyone. I'm Esther Zito. I'm a chief of a laboratory of signal transduction at Mario Negri Institute in Milan. My is an institute of pharmacology, and my lab is focused on the understanding of the biochemistry, the biochemical basis of SELENON or SEPIAN1 related myopathy. So thank you for this invitation.
And I'm Casie Genetti. I'm a genetic counselor. I work with Alan and Behzad at Boston Children's Hospital. I see a lot of familiar names here. I help to run our kind of patient-facing part of our research, so enrolling participants for our natural history studies and also our diagnostic work for our undiagnosed myopathies. So great to be here and share this all with you.
Thank you all so much. So now we'd like to take a moment to have all of our panelists share a little bit about what they have been what kind of projects that they have been working on related to SELENON. Dr. Beggs, would you like to start us off?
Okay, I'll give a quick overview of my group, but leave some of the more granular details to Behzad and Casie when it's their turn. And let me start by saying that the SELENON field, this is obviously quite a rare condition, not an enormous community out there. We're always trying to grow the community. So we're thrilled to have all the participants we have with us today. But as researchers, we've relied a lot on interactions and collaborations between ourselves and with each other, and also collaborations with the patient community. So a lot of what we're doing is actually built off of work that Dr. Ferreiro's lab has done or Dr. Zito's lab has done. So broadly, we are focused on trying to develop therapies for this. And to do that, you need to understand the basic biochemistry, as you'll hear from Dr. Zito, and the pathophysiology Dr. Ferreiro has been working on. And to do that, we have developed a mouse model. There's also a similar mouse model that was developed in France. And these mice have a very subtle phenotype. One of the challenges with this condition is that the animal models that we make, the animals are, it's very difficult to elicit a phenotype, we say, to see what the effect of that genetic defect has on the mouse. But we have mouse models. We've also developed zebrafish models and can show that if we stress the zebrafish, for example, by making them swim in water that's more viscous than normal, that then you can see a defect. And so we've also worked a lot with cellular models based off of data from Dr. Zito and Ferreiro's groups, and are developing methods to screen for small molecules. Another area we're working on is gene therapy. I'll let Behzad tell you a little bit about that. More recently, we've been working on looking at the function of mitochondria in cells and in tissues as a view into what's wrong with the cell and how we might use that as a tool to screen for either drugs, small molecules, or show an impact of gene replacement therapy. And then finally, when Casie speaks, she'll touch a little bit on how we try to identify families. And you can do a mathematical calculation. If you look at the genes of normal, healthy people, and you count up how many people carry one version of an abnormal SELENON gene, you can predict how many people should be present in the population who are affected by virtue of having two abnormal copies, one each from their mother and their father. And what we find is that the number of community members we're aware of or people who enrolled in either my lab study, in the PRCMD registry, for example, as well as in studies in Europe, it's not nearly as many people as we predict should be there. So one of our important goals is to try to find the people who may have SELENON myopathy and yet not know it, not have been diagnosed, not have come to attention. So we can talk a little bit about that. But I'll stop there and we'll let maybe Dr. Ferreiro go next.
Well, thank you, Alan. I think I will expand a bit on what you said. So what we are doing goes from the, as I like to say, from the bedside to the bench and back, because I am both involved on the clinical aspects of this disease in my clinic at the Institute of Myology in Paris, and on the more mechanistic and therapeutic research in the lab. So I just wanted to mention briefly three of the main aspects that we are trying to develop now. First, it's understanding and putting numbers on the way the disease evolves without treatment, because one of the challenges with this disease and with many congenital myopathies is that evolution is very slow, and to be ready to test any treatment is what you will discuss in the clinical trial readiness session. We need to have numbers to measure how the disease evolves in the absence of treatment, to see if the treatment makes any difference. So this is what we do with natural history studies, and recently we have collected a relatively large series of European patients with selenoprotein N myopathy, 132, I think, and we could analyze a series of data and how the disease evolves in the absence of treatment. And there was a simultaneous publication of another series by our UK friends and colleagues. So I think this helps us, this will help us move on to the next step towards clinical trial readiness. Another aspect that we are very interested in is what was already mentioned by Dr. Beggs, and something we discovered with together with Esther Zito's lab, which is the metabolic aspect of disease. We have seen that when selenoprotein N is not there, the mitochondria, which are the energy powerhouse of the cells, of the muscle cells, but of all the other cells as well, do not work properly. So there's less energy generation, less fuel for the cells to work. And this can explain, at least in part, the fatigue, which involves muscles, which, as many of you know, can be the most limiting aspect of the disease in everyday life, or one of them. And that can also affect the respiratory function, because this fatigability is something that we see in the diaphragm as well. So this is an important thing to understand in terms of impact on weight, on body mass, on how this can be influenced by diet, and how this can be restored or improved with drugs to improve this fatigue. So this is something we're very interested in. And the third thing is something based on what Dr. Beggs said before, that the animal models that we have right now have limitations. They have virtues as well, but they also have limitations. So, and in particular, they can't reflect the variety of mutations that we see in patients. So in our experience, cells derived from patient material, so cells from skin biopsies or cells obtained from a muscle biopsy, are useful tools for understanding the mechanisms of disease and for testing drugs that can change this mechanism. And so we want to build up on the experience that we have developed over the years in using these primary cells, which again have advantages and limitations, but we are optimizing them now to use them to try to identify and test therapeutic drugs, but to do it through high throughput screening. That is, instead of testing one drug and then another drug, as we have been doing so far, to be able to use these cells to test libraries, that is collections of 100 or 200 drugs at the same time. And we are interested in drugs that are already on the market for human use, that are already FDA approved. So this is something that is currently going on in our lab, and then subsequently the selected drugs will be tested on the animal models. Another aspect that we are working on together with Esther Zito's lab as well is testing drugs that we have already selected based on the defects that we have identified. We know that these selected drugs can target the defective mechanisms that we have identified. So we are testing them in our models as well. Maybe Esther will talk more about it. And then the third thing is something that is again close to my heart and an endless source of hope and struggle, but we're getting there. As some of you know, we did a small pilot clinical trial with 6 selenoprotein and mutant patients using the antioxidant N-acetylcysteine. Sadly, in the last year with the pandemic, all the clinical research the clinical research teams that were working with us for analyzing this trial were completely taken over by the COVID clinical trials. So this brought our small pilot analysis to a full stop. But the good news is that we have been able to start working on it again for the last 3 months, and I have just received 2 days ago in my email the results of the biomarkers that we were analyzing in this study. So I think this will be important, because whatever the results regarding the effect of NAC, this trial will give us a good idea of the parameters of the things that we can measure and put numbers on, including some what we call biomarkers, that is alterations in patients' blood samples that reflect the degree of oxidative stress, which is a consequence of the absence of selenoprotein N, and also other parameters. And we will be able to know which ones of these we can use to measure the impact of drugs in the future. And also maybe to join the efforts of Dr. Beck's lab in understanding through the registry and through the studies that are going on with the Cure CMD community, how we can bring together these markers of oxidative stress and the lifestyle parameters and the dietary parameters, and how we can bring everything together to come up with more targeted and more effective therapeutic approaches. So all these efforts, of course, are based on collaboration with all the other experts in this rare disease, but not that rare. We estimated from our natural history studies that it's 1 in 1 million people in the general population that are affected. So as Dr. Beck said, more than we know of. And they are based also on the support from Cure CMD, both from the patient community and from the funding point of view. So I will stop here, and just to say I'm very happy to be discussing this with you today, and of course looking forward to.
Esther, would you like to share what you've been working on?
Yeah, sure. So probably if I can, I have also a presentation, just because I have this one.
You can share your screen.
Yeah, I don't know if I can. Only host. Only participant. Okay, doesn't work. So it doesn't matter. So, okay, I want just to update you on the small step that my lab this year has done to the comprehension of the SEPIAN1 function. So we have seen that SEPIAN1 is a type 2 membrane protein, so of the endoplasmic reticulum. So in other words, there is a very small tail of amino acid on the cytosolic side, just one membrane domain, transmembrane domain, and all the rest of the protein, including 2 functional domains, is a calcium binding EFN domain and a pterodoxin-like domain inside the endoplasmic reticulum. We saw that differently from the majority of proteins inside the endoplasmic reticulum, SEPIAN1 is not only a calcium buffering protein, but is a calcium sensor. So in other words, through this calcium binding EFN domain, SEPIAN1 can sense calcium fluctuation. Okay, nice. This is really nice. So SEPIAN1 can sense calcium fluctuation inside the endoplasmic reticulum, and when calcium level get low, SEPIAN1 through the redox domain, this pterodoxin-like domain, can activate a Circa pump that is described as a Circa pump, that is a protein that is able to pump calcium inside the endoplasmic reticulum. This connects the activity of SEPIAN1 with the mechanism of excitation contraction coupling in the muscle. Moreover, we saw that the mutation closer to the EFN domain that are also present in patients affected by SEPIAN1 related myopathy makes SEPIAN1 insensitive to calcium levels. So in other words, SEPIAN1 doesn't sense the calcium, the difference in calcium level inside the endoplasmic reticulum, and cannot activate SERCA in pumping calcium in the endoplasmic reticulum. So we also saw that the lack of SEPIAN1 by disturbing the calcium level, by disturbing the calcium levels inside the endoplasmic reticulum, activates, triggers stress of the endoplasmic reticulum. And we know that this kind of stress can be druggable. So one of the goals of the lab is try to test together with Annette Ferreiro small molecules to understand if we can cure, we can rescue some of the phenotypical aspect of the SEPIAN1 related myopathy. I'm sorry for this strange screen, but there is something working on my computer. Thank you for the kind invitation and all of you for the attention.
Thank you so much, Esther. Behzad?
Hi again, everyone. Thank you for the opportunity to share some of our work here. So I just wanted to confirm what Anna said, that this disease seemed so rare to us back then, 20 years ago, but I'm happy that it's been recognized more and more. And in the last 10 years, especially, patients are being diagnosed more frequently and more easily. So that's really encouraging. In terms of what we do in the lab, so this is probably valid for most disease. So we need to identify the pathophysiology involved. And Esther Zito's group has done a great job understanding what the protein does and how the defect affects the rest of the cell and the muscle. So we are kind of building up on that knowledge developed by Anna and Esther, and trying to develop, first of all, some cellular assays. So the cellular assay, the good thing about that is that you can do high throughput screening. As Anna said, you can, instead of looking at the one or two particular drugs, you can just screen 1,000 or tens of thousands of drugs at the same time. And it is possible to screen drugs that are already approved by FDA for other purposes. And if there is something that kind of works in the cellular assay that is able to kind of correct the defect caused by SELENON, that would be a good candidate to move it to the next step. And the next step would be to try the cherry pick drugs from the initial screening to test them in animal models that we have. So we have the knockout mouse model and the zebrafish knockout as well. The zebrafish also is a nice model because you can have many eggs. So it's kind of have a good, almost like medium throughput, I would say. And then the mouse is the closest model we have as a mammal. So as it has been said, they are not great models because the disease is not very severe in those models. So we need to kind of challenge them. So for example, this could be extra swimming for the fish. We have them actually swim in like a more viscous solution to make their muscles work harder. And for mice, for example, we can have them swim or run on a treadmill to kind of challenge them to express the disease like more severely. That would be closer to patients. And then another completely aspect therapeutics that you are looking at is gene therapy. So this has been kind of successful in other disease that we have been working on in the lab. In particular, I'm thinking about the mytular myopathy. So the good thing about that other disease was that we had a very good mouse model, so it was really easy to develop that. So we're just trying to use our knowledge from an experience from gene therapy in other diseases and apply it to SELENON. And right now we are developing adeno-associated virus vectors to put a SELENON gene basically in that viral vector that could be delivered to patients. But for now, we are at the early stages to develop that vector and try it in mice for now. And I think I'm gonna stop there.
Finally, I believe we have Casie.
Yes, and it's Casie, sorry. No worries. So kind of culminating all of that, everyone's touched on really the importance of involving the patient community in the research that we're doing. So as I mentioned before, a lot of my work is focused on interacting with families with SELENON myopathy, as well as a variety of other myopathies, to enroll them into research protocols that allow us to collect things like previous medical records or biosamples like blood or pieces of muscle tissue that can be used for assays in the laboratory. So many of you may be involved with our protocol already. For those of you who aren't and are interested, you can certainly follow up with me after this, and I'd love to tell you more about it. But one of the main things that we've been doing is some of that natural history work that Dr. Ferreiro spoke about before. We don't see our patients clinically, so unlike her group, we don't have the ability as part of our study to actually do kind of clinical investigation of our patients. But we do a lot of work with the referring neurologists and collecting medical records and helping us to just get a better idea, one, of the prevalence of this condition, and then how it presents across the lifespan, and just kind of how it can vary from family to family or patient to patient. That work is, as Dr. Ferreiro mentioned, very important in terms of being ready for future clinical trials and getting a better idea of what this condition presents like. And we were actually part of one of those recent publications with the group in the UK to try to get that information out there in the literature, and so that it's available for when we're ready to start these different treatments. So really just a big thank you to people who have been involved in research previously. And I definitely encourage you to do so in the future, whether that's through registries like the CMDIR or through participation in studies like ours or others. It really helps to kind of push our work forward. And I'm happy to answer any more questions about that as well.
Thank you very much for sharing with us, Casie.
Sure.
Now we're going to turn to our the questions that our community has asked for us. A lot of these questions have already been answered by our panelists, but I'll take a moment to go through some of these questions that haven't been quite touched upon yet. So one of these questions asks, there has already been a little bit of discussion already in regards to the metabolic component of a SELENON and the research that goes into it. But this question asks, how does it relate to weight issues and appetite? Do you see our lack of appetite as possibly being adaptive? And have there been any studies on the benefit of certain fruits or foods on reducing oxidative stress on bowel and bowel complication reduced lung capacity?
Okay, I think this is something for Alan's group and for me both, and maybe Esther for the mechanistic as well, but it's more like, sounds more like a clinical question. So Alan, is it okay if I start answering?
Yeah, please discuss this at the physiological level first. Yeah.
Okay. So what the collaboration with Esther Zito's lab allowed to disclose is that selenoprotein and presence of a normal or functioning selenoprotein N is necessary for the mitochondria, which, as I said, are the powerhouses of the cell to work properly. And when this doesn't happen, there are two possibilities. First, it can be restricted to the muscle, but it can also happen in other cells in the body. And we looked into this and we saw that this poor production of energy is something that happens not only in the muscles, but also in cells from the skin from patients. So we can imagine, we haven't tested other tissues, we don't do biopsies of other tissues systematically, but we think that the absence of selenoprotein N is not a purely muscular disease. Selenoprotein N related myopathy is not purely a myopathy. It can be more of what we call a systemic disease, that is the disease that touches different systems in the body, and that this has impact on how the body mass is regulated. So this coupled with the clinical history studies, we tried to understand, because weight is a major issue for patients and for their physicians. And very often I have come across patients with very, very low body weight, typically around 35 in adults, who are extremely under pressure because their physicians, their GPs, understandably are concerned and want to help them gain weight to get them closer to the normal standards of weight. And what we have seen is that when they are given hypercaloric foods in the absence of malnutrition indicators, of course, if there is a malnutrition situation with blood indicators of malnutrition, that needs to be corrected. But in the absence of this, gaining weight doesn't necessarily bring them an improvement. And many of them have experienced decrease in their functional abilities when they gain weight through a hypercaloric diet. So what we think after bringing together this clinical study, where we saw that although a majority of patients have very low body weight, there is a minority of patients who are overweight from childhood, and they have a particular aspect with around the belly area. And we saw that these patients who are overweight tend to be on the more severe side of the spectrum of disease severity and tend to lose the ability to live, whilst those with very low body weight have more chances of being in the milder end of the spectrum. So this is an observation. We don't fully understand the causes of this. But what we can say now safely is, I can't say if the loss of appetite is an adaptation response. Probably the loss of appetite is due to a combination of things. Being in respiratory insufficiency will bring your appetite down. So the first thing with a very poor appetite is to check that ventilation is good and that the levels of oxygen and CO2 in the blood are correct, are good. Another reason for poor appetite is the fact that a very full stomach will make breathing more difficult. So many patients have trouble breathing or feel more fatigue after a meal, so they tend to eat less. So in this case, what can be done is fractioning meals, having smaller meals more often, more frequently over the day. But it might be, and I think this is the case, that having a low body weight is part of the body's response to this metabolic situation. And that trying to counter this by forced feeding or by hypercaloric diets from observations, we have seen that it does not necessarily bring a benefit. And there's reason to think that it might not help with this metabolic dysfunction. So checking the breathing, trying to fraction meals, and making sure there is no malnutrition situation. If all these three are okay, then I would say, and the weight is stable or still going up, even though it's slowly in children, because children are growing up and they still need to gain weight, even though the normal weight curve by no means a reference for them. Their normal is different from the population normal, but we still like them to gain weight, even if it's very slowly. If all this is happening, I'm not concerned about a very low body weight in this condition. It tends to be associated with less risk of a severe form of the disease.
Thank you, Anna. Alan,
But
do you
again,
have
this is an
something to add?
observation. We don't have all the explanations.
We have a couple of minutes left. On.
1 or 2 minutes. So actually, Jackie Prostapa, who was with our group, actually sent around a community questionnaire. I'll let her say just a couple of quick words about that. Essentially, trying to measure what people's diet and intake were to try and provide some quantitative data to support what Anna was just talking about. Jackie, you want to say a few words? I think, Justin, she may need to be given permission to unmute.
Yes, sorry, 1 moment, please. Jacqueline, correct?
Yes. Thank you.
In the meantime, because we are very short in time, I want to thank you, all the panelists and all the community that is there, for being here, and we can continue this conversation. You have time to send more questions during the days following, and we will try to address all of them.
And we'd also like to direct you to our community lounges to continue this conversation that we have had during the session as well.
Is Shaki ready?
Yeah. So hello, everyone.
Hi.
I worked with Alan in his lab, and together with the Beggs lab and Cure CMD, we worked on creating a patient reported outcome survey, where we looked at how diet could possibly explain variation that we see in SEPT1 patients, because we have noticed that even people with identical mutations may be affected differently. So we were able to present that data at the World Muscle Society 2020 as a virtual poster. And we're still working on asking the question and understanding exactly what diet can do and can influence in this disorder.
I'll just say the biggest flaw with the study is small numbers, and that's always a challenge here. And so actually perhaps an outcome of this session, maybe Anna, Esther, and others, we should talk about it, modifying if you felt necessary and expanding the reach of that, or maybe doing a second generation that could be a little more meaningful. So thank you, Justin. Gustavo.
I want to ask about one question that we always receive from the community. Are you guys working on gene therapy?
Behzad is developing. Yeah, we didn't go into any of the data. The challenge there is that while the gene protein itself is small enough to fit into AAV vectors, when you include some additional sequence at the tail end of the gene that's necessary for the protein to be made, it's a little bit too large. And so he's developed smaller versions that still work and can fit into a vector. And so those vectors are being produced as we speak. So it's a promising approach. I think one concern, and actually something I want to talk with Esther and Anna about, is could something bad happen if you have too much of it? Because the biochemical work has shown that the amount is very important, and it has to be in balance with other things. So that's something we'll need to address, but we should be able to do that soon.
Thank you. Thank you very much.
So much to our
We are out of time, but thank you, everyone.
Thank you, everybody.
Great to see everybody. Thank you.
Bye.
Everyone.